Thank you to everyone who is still here at the end of the last day of the conference. I know it's a bit of a slog at the end here. So my name is Devin. I'm a postdoc at USGS No-Rock in Bozeman, Montana, and I have been most recently been working with Adam Sepulveda on alongside his ReadyNet project. So I'm going to be talking a little bit about that today. So as we all know, invasive species can be highly detrimental to native communities by outcompeting native species, disrupting food webs, and many other mechanisms. And we know that the longer an infestation goes undetected and untreated, the more likely it is to establish, spread, and cause harm to those native ecosystems and potentially cause losses to valued species in native ecosystems. So as a species becomes more established and spreads, we know that the management cost associated with that infestation increases substantially, and the efficiency of management decreases along with that increase in cost. And it's because of that, investment in early detection and rapid response is likely to have a really high return. And so, that was in English earlier today, I swear. So as a consequence of that, the Department of the Interior, which is a U.S. federal department that houses groups like USGS and National Park Service, has bipartisan funding, which I know is shocking in today's political climate, to improve surveillance and detection of invasive species, which is what that bubble should have said. And so ReadyNet, I know Adam talked a lot about this yesterday, so I won't go into too much detail. But one facet of what they're trying to do, or this program is trying to do, is optimize sampling so that we can tell managers when to sample, where to sample, and how best to sample. And that's primarily focused in on targeted surveillance right now. So as a part of that, trying to find that right tool at the right place and the right time, we're working with the Monterey Bay Aquarium Research Institute to develop these environmental sample processors, or ESPs, or just robots as I like to call them. And they're able to sample from our water sources autonomously, so we can deploy them for months at a time and leave them there to sample at some frequency that we tell them to. And so as we are hopefully finding these right tools that we can start implementing into our eDNA surveys, there are a lot of key decisions that we have to make along the way to ensure that we're getting good data from these interesting tools. So we want to make sure that our data are comparable downstream. And so for my research, I am leveraging existing sampling schemes by the ReadyNet program for targeted detections, but I'm actually using broader surveillance, so using metabarcoding of these eDNA samples. And so the way that these ESPs work, we can deploy them in USGS stream gauges, as pictured here. So this one's out in the Grand Tetons National Park, which is hideous, as you can see. And we're able to put these very large robots, so you can see from here, they're quite large and a bit burly. But we put them in there, we can hook them up to an existing power source or solar panel, and then there's a tube that runs from the house into our water source, and it can pull water from our source of interest, and then it's able to push water. So I have a video here, let's see if that works. So you can see that a puck holding a filter can come up, and then this arm brings it here, and the machine is able to push the water through the puck. And then once it is done filtering at whatever volume of water you want it to, it's then stored in RNA later, and you can come back and pick it up whenever you want to. One challenge with these robots is that they can only hold 132 samples at one time. So we need to figure out how we want to use those 132 samples in an optimal way. And so that brings me to my main questions. So the first two questions are more about the how do we sample. So how does filter pore size affect our detections of fish, and how does primer choice affect our detections as well? And then the third question is really more about that, when do we want to sample? So what temporal scale matters when we're trying to detect these fish? And so we were able to borrow 2 of MBARE's robots and deploy them at 2 USGS stream gauges. One in Jackson, at the outflow of Jackson Lake, right here, which is this one pictured. And then we had an additional one at the outflow of Palisades Reservoir in Wyoming. And this is all in the upper Snake River of Wyoming. And so they were deployed for both 2021 and 2022, and just for July and September. And we picked these sites because, of course, you can see there are some differences, but they are quite similar in a lot of ways. So we see very similar patterns of discharge throughout the months, and similar temperature patterns as well. So even though our Jackson Lake temperature profile is pictured here, Palisades displays similar temperatures, just a little bit cooler, very similar. They also have fairly similar fish at both of these sites, so there's quite a bit of overlap. And we know a lot about the fish that are present at these sites. So this is all from captured data, although there are some fish, of course, that are present only at Palisades and only at Jackson Lake. And as I mentioned, I was interested in different primer sets. So we used samples from 2021 for metabarcoding, and we noticed that there were a lot of fish species that were absent that we should have been detecting. And so in addition to the MyFish primer, we also added this ACMDB primer from Bauman's et al. 2018. And these are both 12S primers, but getting different regions in 12S. So going back to our first question, how does filter pore size affect fish detections? Oops, sorry about that. And so what we see here, so these are samples only from 2021 and amplified with the MyFish primers, but basically we see no difference in within sample diversity or alpha diversity between our different filter pore sizes. The only caveat here is that we were only able to do this with a subset of 19 samples, which is about 6 per group, and we were not able to collect these samples simultaneously. So we had to collect one and then another and then another. And I'll talk about that, the potential effects of that in just a bit. And then second question, back to how primer choice affects our fish detections. So what we see here, perhaps not very surprisingly, our 2 separate primer sets were able to detect slightly different patterns or slightly different fish. And really what we see is that the addition of the ACMDB primer really only added 1 additional fish detection that our MyFish primer could not detect. And so not surprisingly, when we compare alpha diversity between the 2 primer sets, we see that MyFish has a slightly higher alpha diversity when you're looking at either Shannon diversity or species richness. So it didn't really necessarily warrant adding that second primer set, at least for 12S. Perhaps a different region would have been more useful. And so then finally, we want to look at the temporal scale. That matters. So yeah, as Adam mentioned yesterday, this ESP can sample essentially whenever you wanted to. Could it be hourly if you wanted, once a day, 7 times a day, or once a week, etc. And so this is just one example of one fish that was detected, Prostopium williamsoni, which is the mountain whitefish. And so it's a bit of a complicated figure. So we have our Jackson Lake site data at the top, Palisade Reservoir at the bottom. We have our 2021 data on the left, and then 2 columns of 2022 on the right. And then you can see which primer set at the bottom. But basically, I wanted to see how the number of reads for this fish varies over time. So are we seeing really high numbers of reads in the afternoon or not? And basically what we see is that it is highly variable. Oops, sorry about that. So even between months, so these different colors are the different months, you see there's a lot of variation in the number of reads that are detected throughout a single day. So each line represents 1 single day, and each point is a sampling point. And so then if we aggregate all of our data together, I was really curious to see if certain species were more likely to be detected at certain times of the day or not. And so here we have our species that we detected on the left, and then across the bottom we have our start time of sampling. So that would be if we started sampling at 12 a.m., that would be 0. And then the colors that will pop up were the number of days that a fish was detected at that time of day, with warmer colors being more detections. And so essentially what we see is that there are more detections in the afternoon compared to morning hours. And we have a few hypotheses as to why that might be, but we haven't explicitly tested them yet. So that's something that's still in progress. And so if we look at 2 example fish, some of them are only detected at certain times of day, whereas others seem to follow this pattern more clearly with those greater afternoon detections. Okay. And then if we take a few steps back to look at seasonal differences, we find that there are no differences in the alpha diversity or within sample diversity between July and September, which were the only 2 months that I looked at. And that could potentially be attributed to some of the changes in fish ecology. So we might have some fall spawners that are more active in September, like lake trout, for example, or Yellowstone cutthroat trout have like a spring or summer spawning time, so we might see more of them in July. But what we did see was that there was differences in beta diversity or community composition, which is that between sample differences. So even when we account for different primer sets, which is why you see such separation here, we do see that season does change the community composition. And when we look at the data as a whole, we see that our primer site and our year can all have a large effect on what we infer for our community composition. So if we want to look just at our 2 different primer sets, so we have our same 2 sites, Jackson Lake and then Palisades. So these are the exact same samples compared or sequenced just with different primers. But we see substantial separation in the community composition. And then similarly, if we want to look just at the site level, even within years and within primers, we see differences between sites, but that's not really surprising given that Palisades has 5 fish that Jackson Lake does not. And then we also see some annual variation in the community composition of these samples. And that could be for a lot of different reasons. There was a very large flood in 2022. That could have had something to do with it. We used different filter types, and we also sequenced them at different facilities. So there are a lot of different things going on. That's a bit hard to tease this apart. Yep. And so with that, basically, what we've found is that our choices along the way can make a big difference in our results and how we interpret those results. So for this case study, for example, filter size was not so important, but primers and time were potentially very important. So it just really highlights the need for more standardization within field and lab pipelines so that we can have comparable data downstream. This is very much, these are results in the work, so we're still analyzing them. We'd like to incorporate some environmental data to kind of get at the why we're seeing these patterns, especially with time of day and things like that. So yeah, I think that's perfectly on time.